Hello, everyone, and thanks for being here today. I'm glad to present FCS Express to you. My name is Andrea Valle. I'm Technical Application Specialist at the Novo Software. I'm based in Italy, in Central Europe, so I'm the closest of the Novo Software team to you. And what I'm going to be doing today is to give you an overview on FCS Express 7, so our software. I put together a few slides at the beginning to give you an idea of the company, the Novo Software, and then I put together some example of analysis that you can perform with FCS Express. Again, I want to mention that you have a question section in the GoToMeeting dialog. So if you want to type any question during the presentation, I will take care to answer you in real time. Okay, so the Novo Software is a US-based company. We are based in Pasadena here, very close to Los Angeles. And we have very large network of distributor and sales company. So in the company, we are 27 people that are specifically devoted to develop FCS Express 7. We are not doing anything else. And among these 27 people, we are 10 application specialists that are spread in 5 different time zones. As you can see here, all those yellow triangle are application specialists. I'm supposed to be this one. So this is really great to support our customer because we can reply to their email in like 2 or 3 hours. So we are pretty fast. And as I was mentioning, we have a very large network of distributor that we work with. And of course, today I want to mention Merkel technologies that help us a lot to organize this session today. So I want to say a big thanks to all their team. FCS Express, born in 1998, together with the Novo software. When the first version of the software actually was released, thanks to the current president, who's named David Novo. And today, what I'm going to present to you is FCS Express 7. We just released it in August 2019. So in this 22 here, we really work hard to implement new features in the software, and we introduced a few different editions. Again, we are only dealing with FCS Express. So but we introduced different editions. For example, as you can see here in 2004, we introduced the IVD version, so in vitro diagnostic for people dealing with clinical sample and people that need to perform diagnostic. And of course, we have RUO, so research use only version for academic and company that do not perform diagnostic. And we also have a version for imaging that we introduce with version 4. Again, the software is identical, but can deal with multiple file formats. We will mention more detail about this later on. Maybe some of you never heard about FCS Express, can be. With this chart, I just want to show you that our customer are really increasing a lot. You see it's almost like exponential growth. This is actually the number of publication. You can retrieve this data by just going to PubMed, for example, and search for any paper that's cite FCS Express or the Novo software. And this is just to let you know that our customer are really happy, and actually they publish even in high impact journals. So FCS Express is really helping them in their daily research and job. So I would say that we have half customer in the diagnostic field and half customer in research. So we can cover both needs for both those customers. I mentioned that we do have a version called IVD, which is for people dealing with diagnostics. So I'm not sure today the audience is pretty heterogeneous, so maybe some of you is actually dealing with diagnostics. So I want to just spend a few words with this slide on the IVD version. So the IVD version is fully validated. It's listed with the FDA as a medical device, and we are actually open to regular inspection by either the FDA and by the California Department of Public Health. But what I would say is the major point of the IVD version is that it's fully validated. And this means that every time that we release a new update, we have to run thousands of test cases to ensure the quality. It means that we need to check that all the statistics are calculated in the right way, that all the plots display your data in the right way. And this is something unique. And it's really a good point to ensure the quality of your analysis. And I have to mention that internally at the Novo software, we are running all those test cases also on the RUO version of the software. So it's not mandatory, but we are doing that. Because we really care about the quality and the analysis of our customer. And again, this is something that the majority of other software do not have, because most of them do not really have an IVD version. But we do, and this is why we'll also take care of the RUO version too. So before moving on, I just want to mention that FCS Express, it's really a huge piece of software. It's really comprehensive. The manual, it's larger than 2,200 pages. So there is no way in which in like 1 hour I can show you everything. So because of this, I put together some example that I hope will really persuade you that FCS Express is a really great product and that it can help you in your daily job. So the example that I put together are focusing on what I think are the main hurdle in the daily practice in flow and image cytometry. So like, for example, presenting data, managing statistic, high dimensional data analysis. And overall, I would actually summarize all those points in like get the final results. So this is the main point for everyone, not only in flow cytometry at the end. So before moving on, I want to give you a few basic specification about the software that are always useful. So FCS Express run on both Mac and PC, and the software is identical. So we don't have two versions with different functionality, with different features. The software is identical. So you can start your analysis on a Mac, you can move your analysis on a PC and continue to analyze your data. You can go back on a Mac and so on. Then FCS Express, it's compatible with all the flow cytometers available nowadays. We can read FCS file, we can read LMD file. We can also read a bunch of proprietary formats, like for example, we can read files from Sony Spectral, we can read files from Miltenyi. And if you have like image instrument, we can also deal with imaging, as I was mentioning. We have version of the software that can deal with imaging data. And the one that I want to mention is, of course, Amnis. So we can directly import that file for those of you who are using ImageStream. Then a very good point of FCS Express is that it doesn't bring any intrinsic limitation in terms of number of files that you want to analyze in your layout, neither in terms of number of parameters or number of events that you can store in your FCS file. In FCS Express, you can perform, of course, compensation. You can tweak the compensation if you already have a compensation matrix. Like 3 days ago, we released a new update of FCS Express that also allows you to perform spectral unmixing. For those of you who have a spectral cytometer, you can retrieve the standard statistic from your sample, and you can create the standard plot type. So probably I will say that this slide, it's more to let you know that whatever you're doing with your current software can also be done in FCS Express. So what's the point of moving to FCS Express? Or if you just want to try it, what's the point of at least trying FCS Express for your data analysis? And I tried to summarize. So the main advantage in this slide. So this is probably the most important slide of my presentation. It's the take-home message is here. So on the left, you see the standard workflow for users that are not using FCS Express. And on the right, you see the FCS Express way of doing. So let's focus on the left part. So when we sit down with the customer using other software than FCS Express, we always found out that the main hurdles they are facing are not about creating plots and retrieving numbers, like retrieve the percentage of a population. So this is something that you can always do with every piece of software, and I will actually say very quickly. The main hurdles nowadays are moving from those plots and numbers to the final results, because those plot and numbers are not the final results. Even if we say that this is the analysis, actually the analysis, it's a much bigger, let's say, process. Because once you have plots and numbers, okay, then you have to put together your final results by copy and pasting or exporting to PowerPoint or to Photoshop. Let's say, for example, when you want to present your data, when you want to create a picture for the publication, when you want to create a poster for a congress. Similar stuff when you want to deal with statistics. So you need to copy and paste or to export to Excel, to GraphPad, to some statistical software, create a chart, and then of course you need to copy and paste that chart in your presentation or in your poster. And what about when you want to perform some like more complex analysis, like high dimensional data analysis? Well, usually you need to rely on statistical software, or maybe your software can do that, but it relies on plugins that are not always working. Maybe you cannot install them, and so on and so on and so on. So this process, this way of doing things is not it's not efficient. It's time consuming. It cannot be automated. If you have to perform the same experiment twice, you need to go through all these unneeded steps again. And what about if your boss asks you to change a gate in your first part, like in your analysis part? Then you need to do this again, and again. So it's time consuming, it's not efficient. In FCS Express, we try to avoid all of this. We try to integrate both data analysis and data reporting tools. This is why, as you will see, FCS Express bring features, tools like statistical analysis, t-test, ANOVA, McWhinney. It brings charting capability. You can create pie chart, bar plot, box and whisker, regression. You can deal with high dimensional data analysis with integrated algorithms. You can deal with high throughput screening assay or ideally speaking, with any type of plate-based assay. And everything, it's integrated. So if you move a gate, just as an example, if you move a gate, everything will update. So in FCS Express, we'll try to bring you directly to the results. So what I've done was putting together example, and I have three main sections. So the first one is a general example on how you can start your analysis in FCS Express. This will give you the idea of how easy and how user-friendly is FCS Express. And also an idea about how FCS Express can help you in creating like high resolution graphic pictures. The second example, the second part of my presentation is more about statistic and numbers. Not only, and you will see, not really about single statistic like the percentage of a gates, but summary statistic, like when you want to combine and to compare experimental group. And the third part is an example of advanced analysis. So the first example is what you need to do when you want to share your data, like when you want to present your data. For example, this may happen for an informal lab presentation, for a poster, as I mentioned before, for a congress, or even when you want to create a nice and very cool picture for a publication. So The limitation that you will face in other software than FCS Express are the low resolution graphic and the limited formatting capability. So this is why people without FCS Express have to export or to copy and paste in external packages. But this actually introduces, again, a needed complexity. You need to learn other software, and it's time consuming, because if you change something, or if you, in your analysis, or if you do the same experiment twice, or if you decide to use a different sample as a representative sample, you need to go through those steps again and again. What we do in FCS Express, it's really different. So let me start with, you see here, I have a blank layout. I want to start with the blank layout to show you how FCS Express, it's really user-friendly and easy to use. So what you see now on the screen is FCS Express. It's not PowerPoint. But it's really similar to PowerPoint. You see, we shamelessly copy the PowerPoint user interface. And why we did this? Because PowerPoint is really well known. Most of us already know how to use PowerPoint. So this is why we copy the PowerPoint user interface. We have a ribbon bar at the top of the screen. You see here we have different tabs like File, Home, Insert, Design, exactly as in PowerPoint. We have a blank page in the center. Of course, you can add multiple pages by just clicking on this little symbol here. And we also have the zoom. So it's not only similar to PowerPoint, also the way, as you will see, in which we are going to create the analysis is the same way in which you usually create a PowerPoint presentation. So we will add here some title, we will add some plots, we will add some table with statistics. You will be able to add a chart here. You will be able to make calculation with an integrated spreadsheet, calculate the statistical significance, and so on. And the beauty is that what you will create here will be your final results. So you don't need to put together the final results. Okay, the layout that you create step by step, it's already the final result. So what you see is what you get. It's very straightforward. Of course, you can also place object out of the page, as you will do in PowerPoint. You can do that. Just know that if you hit print, the object inside the page will be printed and the object outside of the page will not. That's the only difference. So the first example that I want to show you is that I know most of you probably never used FCS Express. And what about if I ask you, how would you print in FCS Express? So probably now that I told you that it's really similar to PowerPoint, you will just look for the File tab and you will grab the Print button here. You see exactly the same dialog that you can find in any Microsoft Office package. So again, the idea is that the software, it's really user-friendly. If you know how to use PowerPoint, you're probably alpha way of learning FCS Express. So no need to start from scratch. The learning curve with FCS Express for user that are getting started with it, it's really fast. Now, again, I was mentioned that the way in which you create the analysis, it's really similar to the way in which you create a PowerPoint presentation. Let me zoom in a little bit. In PowerPoint, if you want to create an object, you go to the Insert tab. And in FCS Express, you do exactly the same. You can add shapes, you can add text box. For example, you may want to add a title. You see here, you can resize the text box, you can move it around. Here, we also have a text tab, and you see you can change the font properties, you can change the alignment, you can add bullets, you can insert table, and so on. But in the Insert tab, you see we have many other objects. Like, for example, we have plots. Of course, these are not available in PowerPoint, but in FCS Express, you can create plots very easy by just selecting any plots you want, draw it on your page, and then the software is asking you which file you want to load. I will just load one of my file. And this is my plots. You can see it sounds like a picture, but it's not. So it sounds like a picture because, for example, you can resize it, you can move it around. but it's not, because if you click, for example, on the axis title, you see you can access any of the parameters available in that files. If you have like 20 parameters, you will see here 20 parameters. You can add as many plots as you want. Like, for example, I'm going to open a contour plot. I'm going to open a color dot plot. And exactly as in PowerPoint, you can multiple select them, move them around, resize them all together. Here in the Home tab, just to show you, again, I want to reiterate that it's really similar to PowerPoint. We can arrange, we can align, we can make them, for example, the same width and the same height. We can equally space them, for example. You see here, distribute horizontally. We can align them at the top. You can do all the stuff. And of course, you can also place them as you want manually. I want to show you that we also want to make your life easier, not only by making FCS Express similar to PowerPoint, but also by adding a lot of dragging and dropping capability. So the first example of drag and drop capability is how to create a new plot from a gate. So first, I want to show you that we have a gating tab, and that you can grab any gate you want, for example, an ellipse. You can draw it on your plot. And you see here, we let you decide which name, which color, whether you want to include or exclude, and so on. Once you have done, you can just click OK. So this is my gate. I want to point out here, you see this is a color dot plot. You see we are backgating automatically. It's very easy. And as I was mentioning, I want to show you how you can use drag and drop to create a new plot. So it's very easy. I will just drag this gate and I will release it out of the plot. So if I'm going to do this, you see I have a new plot, and the new plot is automatically gated and it's live update. Any single tiny change that I'm going to do will affect my entire analysis, my entire layout. So even if I have 100 plots on 10 pages, everything will update immediately. And if you drag and drop a gate on top of an existing plot, and this is what I'm going to do now, I will drag and drop this gate and I will drop it on this plot. I can apply the gate very easily. So now gate 1 is applied to the control plot. You see, creating an analysis in this way, it's really, really easy. So let's say that I only have one plot. I can drag and drop, and then I can change, for example, my my marker here. Let's say CD4 and CD8. I can create another gate. I can call it with any name. I can drag and drop, and so on, and so on, and so on. So this is the idea. If you want to retrieve statistic, you can easily right click on a plot and retrieve the statistic from that plot. And again, I want to mention that everything is live update. So you see here, all the statistic are live updating. If you want to change a plot to another plot type, you can do that. No need to recreate it. Like here in the Format tab, we have tons of formatting capability. Like this is just one, change plot type to something different, like for example, contour plot. And you see now that I have a contour plot. But you have many. You see here, you can, for example, change the fill. And now, for example, I'm using a gradient from white to pink. I can change the axis fonts and all the axis options, like the scale here, you see. I can change the range. I can change the title fonts. If I want, this is now bold 16 and it's lime. And now you see that the y-axis, because we select y-axis, otherwise you can do it on the x-axis, change. So those are just few of the customization that you can do on any plot type. So let's say that this is your analysis. You will see you can do much more. I just want to mention, but we will see in a few minutes, you can add also chart here. You can add heat map. But anyway, if this is your analysis, you can use FCS Express to present your data, with the advantage that if your boss asks you, what about if I move this gate here? How this affect all my other plots? Well, that's easy. It's already done. While otherwise, you need to go back, open the layout of your software, change, and then export to PowerPoint, and then go to your boss. But if you still want to export to PowerPoint, that's fine. Remember, here in the File tab, this is similar to any Microsoft Office package. We have export. So for example, in PowerPoint, you can export to PDF. We can export to PDF too. We can also export to PowerPoint. We can export to pictures. You can also define the resolution of the pictures. So if the editor asks you, I don't know, for this journal, you have to upload pictures at a resolution of 600 dpi, you will define 600 dpi, and all the picture that you will export from FCS Express will have that resolution. I just want to show you the PowerPoint because probably is the most used export, and I will save this on my desktop. You see we have a few options here, like for example, the resolution. We can select which page. In this case, I only have one page. So I will go straight. I will just save and open the presentation automatically. And this is the PowerPoint. You see, I have my text, I have my 3 plots. If you have 10 pages, you will have 10 pages. If you have 100 file, you will have 10 pages times 100 file, and so on. And I want to mention that this is a real PowerPoint. So first of all those objects are ungrouped. And you can see that those are high resolution. So those are all vector graphic objects. So you see, this is like 400% of zoom and everything looks great. And if you want, you can also right click and ungroup those objects. And now also the title is ungrouped, and also all the axis label are ungrouped, and also the background is ungrouped. If you want, you can customize this object here in PowerPoint, but usually I suggest to do that in FCS Express directly, as most of the stuff can be done in FCS Express, as we did for this plot. Okay, so this was the, let's say, the first example that I want to mention. The idea is that, again, just to reiterate, in FCS Express, we want to make your life easier. We want to integrate analysis and reporting capability. This is why we have fully formattable text and plots. We have high resolution graphics. We do not like copy and paste in at the Novo software. You see, I've exported in PowerPoint without a single copy and paste. So the idea is that if you don't need to use external software to present your data or to put together the final picture for your presentation, because you can just use FCS Express. You can add text, you can add pictures, the logo of your institution, the logo of your company, the picture that you took from your cells at the microscope. That's fine. So at the end, we want to simplify your life. So reduce the complexity and also reduce the time that you spend in front of the computer. So the second example that I want to mention, that I want to present you, is about statistics. So most of the software most of the flow software are really limited in these terms. So every time that you want to perform like a statistical test, every time that you want to calculate some regression, like the IC50, the titration, or the performance of your instrument, and a Nova or whatever, you need to rely on external software. So this is because flow software are usually really limited in terms of calculation and in terms of graphing. So this is why, as you can see here, you need to go through these unneeded steps. And again, I want to mention that if you change something here, you need to go through these steps again. In FCS Express, we don't want you to do this. This example is a very easy example, but I think it really addressed the point. So in this example, I have only 3 samples. Again, it's very easy. You see here I have 3 samples. Sample 1, sample 2, sample 3. I just imagine they come from a kinetic experiment like 6 hours, 12 hours, 18 hours. You can imagine anything you want, like 3 different experimental groups. And I am interested in 3 different populations, like blue, yellow, and green. And I retrieve the statistic that I want. Now, this is the point where, if you're not using FCS Express, you need to export your data to go through like Excel if you want to make a chart. or to copy and paste, which is even worse. In FCS Express, you don't need to do that because you see here, you can create charts inside your layout and you can grab those charts here. You see bar, pie, box and whisker, scatter, scatter with regression. Those are just 3 examples. And the beauty is that since those charts, let me unzoom a little bit, since those charts refer to the statistic that comes out of my analysis, and since the statistics are live updating based on my gates, also on this gate, of course, if I change something in my analysis, all those charts will update automatically. So for example, if I move this yellow gates, okay, this yellow gates will trigger the updates of all the statistics referring to gate 4, you see here, and all the charts displaying the yellow gates. So if I'm going to do this, you will see that the yellow bar, the yellow line, and the yellow slice in the pie chart will update, and also the statistic in the table. Think about doing this without FCS Express. As soon as you change this gate, even by 1 pixel, you need to re-export, redo the calculation, update the chart, copy and paste the chart in PowerPoint every single time. And you need to do this also if you have the same experiment, if you are going to perform the same experiment the day after. While in FCS Express, this layout is actually a template. So if I want, the next day I have other free sample, I will just load those free sample inside this layout, and all the statistic, all the chart will already be there. No need to recreate all of them. I was saying that this is an easy example. It's a basic example because it doesn't contain replicates. And this is why I put together this second example, which is more, let's say, real life. Again, I imagine to have free experimental group, but actually you can have as many sample as you want, as many group as you want. They can be organized as you want, multiple pages, single pages, it doesn't matter. In this case, I have 2 sample for group 1, free sample for group 2, free sample for group 3. Again, you retrieve statistic, but then you want to create some chart, you want to calculate some statistic because you want to compare group 1, group 2, and group 3. What I've done here was to use the integrated spreadsheet. The integrated spreadsheet, which is this one, is like having Excel integrated into FCS Express. If I open a blank spreadsheet, you see here insert spreadsheet, I can clearly show you that it's like Excel. So hope you can see this. We have columns, we have rows. I can make it bigger. I can manually type as I'm doing now. Let me increase a little bit. I'm not sure you can all see what I'm typing. You see here, if you want to create a formula in Excel, you just start with an equal. And then, for example, if you want to sum, you do this plus this. Okay? In FCS Express, it's the same. So we try to reproduce the behavior of Excel inside FCS Express. But the beauty is to populate the Excel with statistic that comes from your analysis. And this is exactly what I've done here. You see, this is the percentage of lymphocyte from sample 1. This is the percentage of lymphocyte for sample 2. You see, this is 1844, which is this one. This is 1868, which is this one. And I've done this for the second population. I've done this for the third population. I've also calculated the average. If you want, you see the same formula that you have in Excel, equal average B2, B3. I've calculated the standard deviation. So whatever you're usually doing in Excel can be done here with the advantage of the live update. And of course, with the advantage that once you have populated the spreadsheet, you can create the chart as we did before. You see here we have box and whisker, pie chart, we have heat map, we have bar plot with multiple series of data. You see here I'm displaying, as we did before, 3 series of data, the red, the green, the purple, and I have 3 experimental group. I have also the asterisk here that define the statistical significance. So you are the one that can place this asterisk. Usually you type it. In FCS Express, you can add it and make this asterisk to updates automatically based on a rule that you have created. And again, look at this picture, you see all those charts. If I go here, for example, and I want to move the monocyte gate, the purple one, you will see that this calculation will update, this statistic will update, this plot will update, this bar will update too, and here I also have a t-test, this will update as well. So again, think about doing this. You can see it's live updating. It's super fast, and this really gives you a lot of power. The same is true, for example, for the red one, of course. And you can also see, for example, that the asterisk can update in real time. Now it's 2, now it's 1, and so on. And I want to mention that we also have regression. We will see this in a later, in the last example of this section. But you see here I have a regression. So I have different value. For example, I have different time point, like in this case, and I have a statistic of interest, for example, the mean of a given population, and I can draw a regression. We have 19 type of regression. This is just a linear regression, but you can add as many regression as you want. And again, this is live updating. So now it's saying me that my treatment increase the percentage of lymphocyte by 168 per hour. But you can apply this to any experiment you have in mind. So the third example of dealing with statistic, it's actually also good for people that usually deal with plate-based assay, because this is an experiment in which 2 compounds have been, let's say, titrated in order to calculate the IC50. So it's a 96 well plates, as you can see here. In this case, this is just a representation of the 96 well plate. All the well are just black. I don't want to confuse you now, but you will see you can color each well. And this is the scheme of the plate. It's just a picture. It's just a picture that I have inserted by going to insert picture. And you see I have 2 compounds. And for each compound, you see compound 1 and compound 2, I have different dilution in quadruplicates, and the dilution are increasing from left to the right. And of course, the idea is to evaluate how the cells react with the increase of the concentration of my 2 compounds. I also have some negative and positive control to the right and to the left of this plate. So this is what I've done in this page 1. You see just a very easy summary with my gating strategy, with my plates, with my picture. And this is my actual analysis. So I have created my gating strategy. It's a very simple analysis in this case. I just want to discriminate between responding and non-responding cells, but you can have, again, you can have as many plot, as many gate, as many pages as you want. And this is my plates colored based on some statistic. And in this case, the statistic you can see here is the percentage of responding cells. And of course, you see the positive control are red because I've used green to red gradient. And also the percentage of positive cells is increasing from the left to the right for both the first compound and the second compound. So I have extracted, this is just one right click. So it's very easy. I have extracted the actual value for the percentage from each well you see here. This means in well A1, I have 41% of responding cells. In well E1, I have 99% of responding cells. Those are indeed the positive control. And you see, I have calculated, you can calculate any statistic. I've just did something very easy, like I've just averaged the 4 replicates and I've calculated the standard deviation. I've done this for the first and the second compounds. This is exactly what I will do in Excel, but I made it here inside the software. And now I have here my regression. I can calculate the IC50, the max, and the R-square. And of course, if you move your gates here, you will see that everything will update. Okay, you see that my curve is automatically updating. All my numbers will automatically update. So the beauty of this is that if you have like 100 plates in which you perform this experiment, once you have created this template, you will just load 100 plates and the analysis will be performed automatically plates by plates. So this is the idea of this second part of my presentation. Again, with the spreadsheet, we want to let you deal with statistics in a very easy way. We have a lot of statistical tests. We have a lot of formula that you can use. Basically, all the formula that you have in Excel, I would say like 99% of the formula that you have in Excel are available in the integrated spreadsheet. So the idea is, again, reduce time complexity, and it's super important, the opportunity for errors. Because if you do not spread all your data in multiple files, and if you don't need to copy and paste to export to multiple file format, if you have everything integrated into the same file, in this case is the FCS Express layout, it would be more, let's say, it would be less frequent that you do some error by copy and pasting or exporting. So I hope those first two, let's say, sections were useful for you. Like, again, we saw how you can start the analysis, how you can use FCS Express to present your data thanks to the high resolution graphic and to the formatting capability that are integrated. We saw how you can deal and what you can do with statistics, but again, we have really much more. And I want to show you just one example of advanced analysis, but I want to mention that we have a lot. You see here, we just saw one example of heat map, but you can do really a lot of stuff with the high content package. We can deal with large data sets. So we have customers that are dealing with files that contains millions and millions of data and really a lot of parameters. You can do proliferation analysis, cell cycle. Again, I want to mention that if you have amnis, image stream, whatever I showed you so far can be applied with those kind of data set, with the advantage that you will also have access to single cell picture inside the layout. Last but not least, we can deal with high dimensional data analysis. So if you have complex data, if you want to run a clustering algorithm, dimensionality reduction algorithm, you can do that inside FCS Express. So in a very easy and user-friendly user interface, and very quickly. So I want to mention, I'm just going to show you an example of Disney, because Disney, it's really, let's say, diffused nowadays. Everyone wants to run Disney. Everyone asks about Disney. It's just an example. We have Disney, Spade, K-means, PCA, and we will soon release FlowSum and UMAP. So we are always working to implement new and to integrate new algorithms. I want to stress again that all those algorithms are integrated. We are not relying on plugins. We are not relying on external software. And this is why, for example, our algorithms are much faster than in any other package. Like Disney, it's between 2 and 10 times faster than compared with any other flow software. So again, this is just an example in which I've run Disney on a population of interest. You see, you can open Disney in a standard plot. This is a density plot in which I have colored each events in this plot based on a specific marker. So what you see here is a shade of color that really helped me in defining, for example, which are the CD3 positive cells. But if you switch this and you color based on the CD19, you can see that now the CD19 highlands is here. So what I've done, just as an example, was to gauge 3 CD3 positive population, like this one, and this one. I just call them subset 1, subset 2, and subset 3, because at this stage, you may not even know who they are. You just know that they are CD3 positive. Of course, you can change the color here to other marker and to then define which is which. But I want to show you other way in which you can do that. For example, you can see here we have what we call parameter heat map. Let me increase a little bit. I'm afraid it's too small. So this is a parameter heat map. It's very easy to use. You can grab it here, insert parameter heat map. This is one of the newest features that we added in FCS Express 7. You see here I have multiple gates. In this case, I have 3 gates, subset 1, 2, and 3. And I can create an heat map in which I can see the comparison of the expression of multiple markers. So for example, I can see that subset 1, it's negative for CD8 and CD4, while subset 2, it's positive for CD8, and subset 3, it's positive for CD4. Another way in which you can do this, in which you can characterize a population, is by focusing on a specific population and open what we call parameter overlays. So basically you focus on a population, in this case is subset 3, which is this one, which is also this one, the last row. And you can plot in an overlay, sorry, in an histogram with multiple overlay, all the marker that you are interested in. Let me make it a little bit more bigger here. So you see, in this case, we can clearly see that this population, so subset 3, is positive for the PE size 7. But the beauty is that if you want, you can navigate, you see here, through your Disney plot and retrieve information from that population of interest. So you see, this is subset 3 again. So if I move subset 3 to a different location, I can see the expression marker on that island. So in this case, I'm focusing on the bottom left, and I can clearly see which is APC size 7 positive. And if I move here, we already know that those are CD19. You can see that those are CD19 positive. You can see it here. So navigate through your Disney plots and look at either the heat map or the parameter heat map or the parameter overlay. It's a very convenient way to explore your Disney plots. And of course, you can always take advantage of the spreadsheet. You see here I have multiple statistics. For example, in this case, I'm plotting the percentage of these 3 population of interest. And if you have multiple files, if you're analyzing multiple files together, you can retrieve those statistics for multiple files. And then you can also plot box and whisker. So for example, it's a very easy way to have very quickly chart depicting a specific population among different file and groups. So for example, in this case, I have 8 files. They are subdivided, you see here, in 2 different groups. Like this is A, B, B, and so on. This can be like treated control or day 1, day 2, or whatever. You can have 10 different groups, two group, it doesn't matter. And this is my box and whisker. So again, if I move subset 1, you see that the box and whisker for subset 1 update in real time. So again, think about doing this without FCS Express. Okay, so again, we have really a lot of stuff going on, a lot of features. We have no time, unfortunately, to see all those cool features in a 1-hour webinar, but I hope I gave you a good flavor of what you can do in FCS Express. So I want to summarize, and I want to mention again that we found out that what we called analysis part, what we usually call the analysis part, is just a small part of the entire process. Because after creating plots and gates, you need to spend time to report, to share your data, to present your data, to create chart, to calculate the statistical significance between experimental group. And this is why at the Novo software, we focused a lot in making the reporting and the graph generation part shorter. And this is why the time saving that you will have through FCS Express, it's so huge. So what I usually say, what I usually ask to people when I present data is, it's a matter of how do you want to spend your time. So that time that you save by using FCS Express can be used to make more science, to read paper, to create poster and other paper manuscripts, or maybe to go home earlier in the evening, instead of spending a lot of time as I would say a graphic designer, because you need to do tons of copy and paste and customization in PowerPoint or Photoshop. So just to wrap up, with FCS Express, we want to save your time. And when you save time, you also save money because you can dedicate the remaining time to do something different. We want to help you in reducing the opportunity for error by integrating everything in a unique layout, which is able to live update. And we want to make your life easier also because we reduce the complexity, because it takes shorter time to learn FCS Express. It takes shorter time to teach FCS Express. You will have to deal with a single package. You will not have to spend time to learn multiple package, and also reduce the complexity in terms of archiving data, because you will have your FCS file and you will have your layout file. So everything is condensed in our motto, which is turning cytometry data into result, because we want to bring you from your raw data to the final results. If you have any question, again, just send us an email or contact Merkel Technology. If you have any question, they are super glad. They are always happy to help you. We are always happy to help you too. So feel free to send us or them an email. And last but not least, if you're interested and if you didn't try FCS Express yet, just navigate on our webpage. So www.denovosoftware.com and click here, you see free demo. So this is the page that will appear. You can also reach it directly by copy and pasting this. The demo, it's fully functional. It includes all the features. It includes features for flow and image cytometry. So if you have an image stream, for example, or if you have image data, that's fine. It also includes all the additional packages, like the one that I mentioned before, the eye contact, that helps you in dealing with plate-based assay very easily. So again, thanks to all of you. Thanks to for attending. Thanks to Merkel Technology for organizing this session. If you have any question, again, just let us know. Okay, many thanks.